I think the microphone is a weapon. I don't think you guys will have a problem hearing me, right? Can you all hear me well? Okay, and I speak to, I've spoken to 1,000 people without a microphone. So you guys, I think we can do this, yeah. So thank you again for inviting me. It's a pleasure to be here. I'm going to be flying to Sri Lanka after this talk. So, and I did not want to say no to this talk because the chapter, Santa Clara chapter, was really my kind of gateway to California botany. During my time at San Jose State, we did a lot of collaborations. I got to know a lot of local botanists through the CNPS chapter here. So you guys hold a very special place for me. So it's great to be here.
Thank you.
Of course. So I'm a geobotanist, and basically I'm broadly interested in understanding how geology shapes the plant world around us. And I've had a kind of a long-term fascination with plants that grow on rock outcrops. And so today what I decided to do was to kind of share with you some key findings in ecology, in evolution, in physiology, in conservation biology, and restoration, and also green technologies from the study of plants that are largely restricted to rock outcrop plant communities. So a central kind of question in ecology is why things grow where they are, where they do, right? So, and we know that climate obviously is central to understanding distribution patterns in any organism, and obviously in plants as well. But within a climatic region, it's a geology that contributes to habitat diversity and species discontinuity. Alfred Russel Wallace in the 1850s wrote that there's nothing more abrupt than change due to diversity of soil. And that's exactly what you see here on this slide, taken at Jasper Ridge Biological Preserve in the early 1980s, showing a plant, mostly Lasthenia californica, up on the Serpentine outcrop. I guess this doesn't work, sorry, I forgot. The laser pointers don't work on screens like that, but you can see that the yellow is mostly Lasthenia californica, growing up on the Serpentine outcrop. Serpentine soils are generally nutrient poor. There are lots of heavy metals in serpentine soils, so somewhat of a harsh substrate for most plants. On the sandstone, you get a lot of grasses and oaks and other plants, and the boundary between the two species generally stay the same from year to year. So when you do reciprocal transplants, so botanists can move things around and figure out why things are where they are. So when you move the Lasthenias to the grassland, in the presence of grasses, they don't do as well. They get outcompeted by the fast-growing grasses. When you move the grasses to the Serpentine outcrop, especially the non-native grasses, they don't do as well on the Serpentine outcrop because they are unable to deal with the harsh chemical conditions that the Serpentine outcrop soils have. So this shows that the soil conditions do contribute to this interesting pattern of distribution that is pretty sharp across the geologic boundary. But what's even more interesting is what you find on the Serpentine outcrop. I don't know whether you can see that, but you see a kind of a brighter yellow down here than up there, right? So up there, what you have is a different species of Lasthenia that is identical morphologically, more or less, to what's found down there. There are two species in the same genus. Lasthenia gracilis is on the upper reaches of the outcrop. Lasthenia californica is on the bottom reaches of the outcrop. And because of the slope, there's leachate of heavy metals and ions that aggregate down at the bottom swale, making the soils down here much more saltier and has higher pH and organic matter content. So it's slightly different soils that you get down here than you get up there. Area up here is mostly sandy. Even though it's still serpentine soils, they're much drier than the soils down here. So even within the Serpentine outcrop, you have this kind of sharply defined boundary that Wallace was writing about in the 1800s. So the boundary again, at least during the time that I was looking at this outcrop, didn't change much except in years where there's a big difference in rainfall. Generally, the boundary stays the same. These guys are annuals, so they start by seed every year, but by the time they come into flower, they're basically spatially segregated into their respective soil habitats, which I think is pretty cool. So we did a bunch of experiments when I was doing my PhD and master's at UBC. I would come down to Jasper Ridge. There was a trailer down at the entrance. I would stay there, climb up the ridge, and do all sorts of experiments up with this population, So through both field experiments and greenhouse experiments, we showed that Lasthenia gracilis is a much more drought-tolerant species. whereas Leycinia californica is a much more soil-tolerant species. Right? So the two species have different physiologies where that enables them to deal with these conditions that are found on this outcrop. When we do reciprocal transplants, when we move the gracilis down to where these guys are, and these guys up there, they don't do as well. Right? These guys are better fit, or Leycinia californica is locally adapted to the harsh, salty environment at the bottom, whereas Leycinia gracilis is locally adapted, meaning they're better fit in these kind of drier serpentines up on the top of the ridge. We also showed that there is reduction in gene flow between these closely sympatric or parapatric populations. They're side by side, but they flower at different times. So when you're flowering at different times, you have a reduction in gene flow that comes with it, right? Because these guys are always at kind of peak flowering when these guys have already gone by. So there's a limited amount of gene flow that happens across these two populations. We brought them into the greenhouse, tried to cross them, and we've also shown through crossing studies there's a reduction in gene flow because they are largely incompatible. There's some amount of hybridization, but it's largely, mostly, they breed within each other, so there is a reduction in gene flow across these two populations. So as a kind of a geobotanist, I like to think of the world as a patchwork quilt, right? So you have these different substrates that are scattered across the surface of the earth. And in each of these substrates that are chemically or physically distinct, you could potentially have different species that are locally adapted to one condition or another. Right? So when you think about that, the world that way, you could imagine how then you could have these distinct plant communities that arise under different conditions that are characterized by either soil chemical differences or soil physical differences. So my fascination with botany or geobotany comes from being able to study these floras on these different types of rock outcrops that have very, very different chemical and physical soil conditions. I work on guano deposits where the soils are very, very high with nitrogen, most very acidic, sometimes about 3.5 pH, so incredibly harsh conditions for plants to grow. I work on copper and zinc and lead mine tailings across most of the East Coast, but also in some other parts of the world. Gypsum, limestone, dolomite, these are carbonate-rich rocks that have high pH, high levels of calcium and magnesium in them. And of course, my favorite is California state rock, serpentinite, and that's where my geobotanical journey started. So that's always going to be my favorite rock outcrop to work with. You're going to hear a lot about serpentine today, but I don't want you to realize that there are these other outcrops that we in California have to start paying attention to. What's going on serpentine is going on elsewhere as well. Right? But because serpentine has been so well botanized, we know a lot about how plants adapt to serpentine soils and their ecology, their evolution, and issues, aspects of conservation, but the same kinds of things are happening in other harsh soils as well. Serpentine outcrops obviously have been studied a lot, in California as well as around the world. These are some of the kind of the major treatments of serpentine outcrop ecology, conservation, evolution that have come out over the last 20, 30 years. And including this book that Susan Harrison at UC Davis, I'm sure you may have had her give a talk here at some point. She's another serpentine ecologist. She and I edited this book in 2011, inviting people from across California to write about how serpentine can be used as a model system to ask questions that they ask in areas of ecology, evolution, and conservation biology. Of course, my fascination with serpentine had a lot to do with reading articles written by Professor Art Krukeberg. I don't know how many people have been out in the field with Art. Art's an amazing botanist, California native from Southern California, ended up doing his degrees at Stanford and then at UC Berkeley, and got a professor job position at University of Washington, taught there for a long, long time, came up with a bunch of books and lots of papers that describe plant-soil relationships across the West Coast in Western North America. So Art passed away about in 2016, and I had the great pleasure of working with Art while I was a graduate student. He hired me as a teaching assistant a few times, And the first time he hired me, it was to teach geobotany in the Klamaths. I'd never been to the Klamaths, and I'd never stepped on a serpentine outcrop. Right? So I was trying to figure out why he wanted me to TA for him, and I wasn't even from University of Washington. I wrote to him because I was fascinated with his writing, so he invited me to be his TA. Day one, I realized that all he really wanted me to do was to be a designated driver after he was done with teaching, so I could drive him to all of the wineries in Southern Oregon. So I did that for 2 weeks, and so that was my kind of first interaction with art. I got to know him over the years, and I ended up writing his memorial for Madronio. I don't know whether you guys have came across that, but I do mention this little interaction with art in the Klamath Mountains. But art has done a lot for geobotany in California. So serpentine obviously was art was fascinated with serpentine outcrops, and so from what we now currently know, we know that serpentinites are essentially mantle rocks that are found in subduction zones, in areas of mountain building activities. We have serpentine outcrops all along the west coast. Also, people don't know this, but there's a lot of serpentine all the way from Newfoundland down to Georgia. Almost as much serpentine that we have here, we have on the East Coast, except that some of the East Coast serpentine outcrops are covered by glacial till, so you don't have this kind of direct serpentine effect that you get in places like California. But if you ever go to places like Newfoundland, Rosemont National Park, massive serpentinite body. Mount Albert in Gaspé Peninsula has a lot of serpentine. Vermont, Maryland, Pennsylvania, all of those areas have large serpentines, and about 6 serpentine endemics in the East Coast compared to 252 here in California. So the numbers don't quite match up, even though the area that is covered with serpentinites are about the same. So the cool, kind of the harsh aspects of serpentine is that serpentine soils have a lot of heavy metals. Right? Nickel, chromium, most of them are not essential for plant growth except nickel and manganese, and to some degree cobalt. A lot of it's toxic in high concentrations. Serpentine soils have very unusual nutrient balances. Nitrogen, phosphorus, potassium are essential nutrients. Right? If you buy a bag of fertilizer, what you get is NPK. Right? So NPK is very low in serpentine soils. But what we think is the most important weirdness of serpentine soil is that it has a very low calcium to magnesium ratio. Calcium is essential for plant growth. Serpentine soils have very little calcium in them, but very, very high magnesium. So calcium and magnesium are about the same size ion. So what happens is that magnesium then takes over the uptake sites of calcium in the root. So plants can die of magnesium toxicity. Right? So plants that grow on serpentine either can keep the excess magnesium out of the root system, or they have really efficient calcium uptake systems, so they can take up high levels of calcium when there's very little calcium in the soil to begin with. Right? So that, we think, is the primary factor contributing to unusual flora that you get on serpentine soils. They're open. You guys have all hiked around in California. You see serpentine outcrops. They're generally rocky, open, very little soil developed on the rock. So it's dry. So water is hard to get on serpentine outcrops. Due to all of these crazy characteristics, you end up having these communities of plants that grow on serpentine soils that are characterized by high levels of endemism, meaning that largely restricted, and also a lot of rare plants. So serpentine obviously is important to all of us because every time you touch something that's made out of stainless steel, it came from serpentine. Asbestos is in the news all the time. There are various types of asbestos, some not as harmful as others, but asbestos belongs to the serpentine group of minerals that is found in serpentinite rocks. If you travel in South Africa, in Cuba, and parts of Mediterranean Europe, you can buy jewelry and sculptures and all sorts of things that are made out of serpentinite and related rocks. They're easy to work with. And of course, you have people who actually, they basically have serpentine tattooed on themselves. This is the graduate student I met at the California Botanical Society meeting. She comes up to me And the first thing she does is rolls up her sleeve and shows me her arm. You know? And this is Danny Black, who works on Aquilegia columbines that grow on serpentine. She's a graduate student of Scott Hodges at UC Santa Barbara. And this, I've never seen this before. I mean, I've seen everything to do with serpentine. I've never seen anybody tattooed it on them. But that's the minerals formula for chrysotils, which is a type of mineral that you find within serpentine rocks. So this kind of really harsh nature of serpentine soils, you know, contributes to conditions that lead to evolution of new populations and of new species across these kinds of boundaries, right? Because there are adaptations that have to take place to be able to colonize serpentine outcrops, and these adaptations sometimes bring with them traits that contribute to reproductively isolating populations that are across these different geologic boundaries. Right? So all I want you to know here is that there's a lot of genetic work going on right now, right, with respect to understanding how plants adapt to serpentine soils. There are lots of model organisms that people are using, Silene, Mimulus, Arabidopsis. So that work has shown that serpentine tolerance is not just one trait. It's not just one gene. It's polygenic. There are multiple genes associated with serpentine tolerance. But all of these studies show that the key aspects of serpentine tolerance have to do with drought tolerance, heavy metal tolerance, and calcium to magnesium tolerance. Right? So these are the key things that plants have to overcome to be able to colonize and thrive on serpentine soils. At the same time, people have been looking at these populations that are found on serpentine and other soils nearby, and they're finding that these populations are isolated, more or less isolated reproductively by all of these different ways. They flower at different times. There is a greater degree of selfing, meaning that they can, you know, they don't need another plant to set seed. So there's selfing capacity in serpentine plants. Sometimes when you cross plants that are, you know, on populations of plants that are on and off of serpentine, the hybrids are inviable, or they're sterile, or they don't do well on each other's parental habitats. So that can also keep them separate. And there's also pollinator shifts, because when you're growing on serpentine, it's a harsh soil. So plants are smaller. You've seen that if you've ever been on serpentine outcrops, plants are generally smaller. If the plant is small, the flower is going to be small. So suddenly you might have a pollinator shift. Something that was like a bumblebee pollinating a flower on a more nutrient-rich soil might not be able to enter a tiny flower that is growing on serpentine. So that could contribute to kind of a pollinator shift that can happen across this soil boundary. But the coolest thing is a recent discovery by some researchers at University of Pennsylvania that show that heavy metals get into the nectar of flowers on serpentine soils. And that heavy metals can then deter certain types of insects from coming to them, and attract certain types of insects that are tolerant to the heavy metals in them. So through that, you could have kind of pollinator-mediated reproductive isolation that can happen in plants that are right across from each other, but one growing on a heavy metal-rich soil and another growing on a soil that lacks heavy metals, which I think is really, really cool. So California is obviously very exciting, but I just wanted to kind of highlight some of the other parts of the world that if you ever get to go to. New Caledonia has a large number of serpentine endemics. 83% of all of New Caledonia's endemic plants are restricted to serpentine soils. It's a little serpentine island, essentially. Right? There are entire monotypic families that are restricted to serpentine soils in New Caledonia. Also an area that's a lot of mining going on, nickel mining, so they're largely threatened. A lot of the serpentine outcrops are largely devastated through mining operations. Cuba, the same story, a lot of mining going on, but a third of the species endemic to Cuba are also endemic to serpentine soils. Sabah, Malaysia is a place that there are a Dutch and a French group working there, and they've only done like 5-6 years of work, and they've already shown that 5% of the total vascular flora found in Sabah is found endemic to serpentine soil. So these are some of the hotspots of serpentine endemism around the world. I was on Mount Kinabalu, which is one of the highest peaks in Southeast Asia. Part of it is serpentine and part of it is granite. On the serpentine side, you have orchids that are endemic to serpentine soils. It's a 14,500 feet high mountains. So as you go up the elevational belts, you find different orchids that are only restricted to serpentine soils. But the most exciting stuff that I saw had to do with pitcher plants that are restricted to serpentine outcrops, some with pretty big openings. Nepenthes Rajah, this guy the openings can be about this big, right? And these are largely found on the serpentine aspects of this height that you take up to get to the highest peak in Southeast Asia.
So that's the king of serpentine.
It's the king of Nepenthes, essentially.
Yes.
This is a large genus of pitcher plants in the Southeast Asia region. And I've found other pitcher plants that are also restricted to copper mines and zinc mines in parts of Southeast Asia. But Nepenthes Rajah is strictly a serpentine endemic, and it is found kind of mid-elevations on Mount Kinabalu. And definitely, I didn't see anything big, but easily a frog or a lizard or something small could fall in there. It's a pretty big opening. So California is where they live, and of course California is another serpentine hotspot. Statewide, there's about 1.5% of the landscape is covered with serpentinite and related rocks, but 14.5%, I keep doing this, I know it doesn't work, sorry. 14.5% of California's endemics are serpentine endemics, they double as serpentine endemics. So that's about roughly 252 full species. And obviously, all should know that California state rock is serpentinite, not serpentine, because serpentine is a group of minerals. The rock is serpentinite. So California state rock cannot be serpentine because that's a mineral. It has to be serpentinite. So correct people when they keep calling serpentinite serpentine. So, the serpentine right up and down the coast. A lot of the large areas of serpentine is up in the North Coast Ranges, the Klamath Mountains. There's a lot of endemics up there too compared to southern parts of California. So endemism rises as you go up the coast in California. The highest levels of endemics are up in the northern parts of California. Harrison's work suggests that this may have something to do with higher precipitation reached in that particular area, making that area much more biomass rich. So there's a greater productivity that would maybe then push some of these specialist species to localize in harsh environments, because regionally there's a lot of biodiversity. The other reason could be that there's a lot more extensive open serpentine regions, there's a lot more microhabitats for things to evolve in, and at the same time, some of these outcrops may have been exposed for much longer than the ones that we have further south. So these are possible reasons for why we have higher levels of endemism up north than down here. Some of my favorites that I just saw, I was in Medford, Oregon recently, and looking at serpentine plants in some of the Jeffrey Pine savanna areas and fens. And Cypripedium californicum is a California lady slipper. About 85% of the time it grows on serpentine. Darlingtonia californica, which is your cobra lily, beautiful. It's not a Nepenthes Rajah, but it is still pretty impressive. Tall pitcher plants. And they are about 75% of the time that you get Darlingtonias in California, they happen to be on serpentine fens. So fens have contact with groundwater, so they're not exactly a bog. pH is slightly higher than what you find in a bog. So the most recently described serpentine endemic anywhere in the world happened in my backyard. So this is in San Luis Obispo. How many people know about this story? It's big news. CNN, BBC, everybody's known. They're not covering it. But this was discovered by a Cal Poly graduate who works for an environmental consulting firm. Corysanthe affinantha is a spine flower. It's called the Irish Hills pine flower. Was discovered last year and just recently described in Madronio as a new species that happens to be a serpentine endemic, and they are now trying to get it listed because it's only found in that one area called Irish Hills open area, right? And it grows right next to Corysanthe breweri most of the time, which is a plant that you must be familiar with. Much bigger flower. This guy is tiny, right? So, yeah, I mean, obviously, it was overlooked because it looks like it, but much smaller, which are much smaller inflorescence. So the person who discovered it is Kristen Nelson. So this is a close-up of the flowers, the fruits, I guess, and there's some flowers still available. She discovered this as part of a survey she was doing for her environmental consulting firm. Tried to figure it out. It didn't key out to anything. Took it to Dave Keel. I don't know whether you know David Keel. He's been teaching botany at Cal Poly for about 40 years, the local expert, and he looked at it and said immediately, like, never seen anything like this before. So was recently described, and now we are currently doing some genetic work to figure out how distinct it is compared to some of the other local horizontal species that are found in that area. And the other cool thing about California serpentine is that California is the only place in the world that has a serpentine endemic vertebrate. Right? You guys hear about this discovery? Again, big news, right? Fish species. I know we are botanists, but at the same time, we have to appreciate when new things are discovered on serpentine all the time. So this fish was described by Peter Moyle and colleagues at UC Davis as a serpentine endemic fish because it is only found in little pools up in the Red Hills area where the pool is over a serpentinite bedrock, right? So, I mean, you could argue this, you don't really know why, whether it has where there's a reason for that to be, there, or it could just be trapped in little pools that have serpentine to begin with. But it is only found, this is years of work shows that this fish is only found when the pool is on serpentinite rock. So, which is really, what is exciting about it is that no one has ever described a vertebrate that was endemic to serpentine, so we have now one in California as well. So 50% of California's endemic plants are strict substrate specialists, right? So if you take California's endemic plants, half of them are found on serpentine, gabbro, ion formation, carbonate rock, so they are substrate specialists, showing that geology is really, really important to pay attention to when it comes to understanding the endemism aspects of California plants. At the same time, about half of California's listed plants, these are the rare, endemic, endangered, threatened species, are also substrate specialists, right? So substrate plays a huge role in thinking about conservation and diversity in our state. So that is why it's really important for us to start paying attention to substrate-plant relationships when we're thinking about climate change. None of the climate change models we currently have that talk about plants moving north or moving up the mountains, as climate changes, have taken into account their substrate affinities. If you are endemic to a gabbro outcrop or a serpentine outcrop, you cannot just move north. You have to find the gabbro outcrop that might be next to you or could be 100 kilometers up or east or west. Well, not west, but not east, but west, right? That you will have to get to. So your success depends on how quickly, how far you can disperse, right? So you really have to pay attention to substrate affinities when you're modeling for climate change across California and across the world. But it requires a lot of work because I've been trying to figure out substrate affinities and it's not easy. Right? You can get that information from herbarium sheets, you can look at CNDDB databases, but you also have to ground truth. You have to have people in those areas telling you that it's only found there. Right? So it takes a while, but it's an important thing to pay attention to when you're thinking about climate change. Lots of soils in California has endemics on them. Right, this is some of the work that we are currently doing. Nick Jensen, who works for CNPS, and I are going through all of the databases for California plants and trying to figure out the level of affinity some of these endemics to have to various types of soils, including sandstone, verticlay, shale, granite, gabbro, limestone, gypsum, dolomite on all of these substrates to figure out whether we can come up with some kind of an affinity rank for these endemics that are supposedly endemic to one type of substrate or another. So these are some of the plants that we've come up with. I mean, they're not strictly endemic, but they're largely found on various types of substrate, gabbro, limestone, gypsum, laterite, dolomite, granite, and they are largely restricted. This is still preliminary work, but we need local botanists telling us whether what we're finding through herbarium sheets actually is really what is going on in the field, right? So one of the things I wanted to kind of talk about today is this kind of really fascinating aspect of plants that grow on serpentine soils, right? So serpentine plants obviously have to deal with heavy metals. Most of the plants that grow on serpentine soils recognize the metal ions, they don't take it up. Even if they take it up, they restrict it to the root system, they don't translocate it to the shoot. Because shoot is where you have to keep the metals out from, because the leaves have to photosynthesize. There's a lot of interesting, important physiological stuff that happens in the leaves, so you have to keep those toxins out of the leaves. But about 600 plants that grow on serpentine soils globally can do this weird thing by which they take up huge amounts of heavy metals and take them all the way up to their leaves. Right? So these are called the metal hyperaccumulators. Right? And the definition, the cutoff, the threshold for metal hyperaccumulation is that you should have at least 1 milligram metal per gram of dry leaf tissue. Right? So that's the bare minimum. But there are some plants who take up to 25% of their dry weight in heavy metals. And that would kill most of us in this room for certain types of heavy metals, right? So there are these plants that are vacuum cleaners. They just suck it all up and they put it in the one place that you think it shouldn't put it in, right? Which is a strange kind of physiological behavior. So this phenomenon, is something that we know a lot of right now, but it was first kind of described or hinted to in 1583 by this Italian naturalist who said that this Alyssum bertolonii back in the day, now it has changed genus, like we know this is happening quite too often, and some of us who are familiar with the older names want to hold on to the older names, so I call it Alyssum bertolonii. Alyssum bertolonii, he said, for people who are mining for nickel in Italy, he said, if you want to find good nickel mining sites, look for this plant. Yeah. If you find this plant, you'll have good grade nickel underneath it. Right? So it was a metal or a nickel indicator plant. Now we know that this plant takes up about 1 to 2% nickel into their above ground tissue. Right? So that, it's a nickel indicator, but it also has this really unusual physiological capacity to take up nickel, dump it into the leaves. Right? So I was in Cuba about 10 years ago with Art Krukerberg from Washington. He was in his 80s at the time. Bob Coleman, who's a Stanford geologist who's responsible for basically mapping a lot of serpentine across up and down the state. Earl Alexander, who's an amazing pedologist, soil scientist, who's, if you want to know where serpentine is, you call Earl. He'll tell you where serpentine is. And then Dick Walker, who was the first person in the 1950s to point out that calcium-magnesium tolerance is key to serpentine tolerance. So what we know about serpentine now, with all of the tools we currently have, were basically, talked about, like in any other subject. We just always stand on the shoulders of people who came before us, right? They figured things out. We just have better tools to show what they showed us way back when. So Dick has passed on, Art has passed on, Bob is in his 90s now. He still lives in Menlo Park. So whenever I come to California, he wants a Starbucks and a chocolate chip cookie. I haven't seen him in about 5 years. I hope he's still fine. Do you have any news on Bob? Bob Coleman? No? Yeah, amazing geologist. And Earl is in his kind of mid-80s and still traveling the country looking for serpentine soils. So when I was in Cuba, one of the things we found is that when you see plants in the Euphorbiaceae, Phyllanthacea, Myrtaceae, especially euphorbs, you break the leaf, in most cases you get this kind of blue-green sap, latex, that comes out of the cut surface. And that latex is enriched with nickel. Right? So there are 140 nickel hyperaccumulators in Cuba. Right? So and at the genus level, there are 33 serpentine endemic genera in Cuba. And Cuba is, serpentine outcrops are not pristine. Very few of them are protected. There's still active mining going on in some of these sites, right? But a lot of biodiversity on Cuban serpentines. But the discovery of this phenomenon happened in the 70s when accidentally a person that I've got to become friends with fell and grabbed onto a plant in New Caledonia. It broke off. And most of us would have been happy that we didn't go down the cliff. But in his case, he immediately saw this blue-green latex coming out. He collected it, took it to his lab, and found out that 25% of the dry weight of this latex was nickel. right? At the time, this actually ended up in Science, it ended up in a bunch of popular media sites, and when they went back to collect this plant, in the Serpentaceae, they found that every part of the plant had more than that cutoff point for nickel hyperaccumulation. So we now know that most of the nickel either ends up in the latex, which is something that plants generally use to defend themselves against insects and pathogens, or they put it into the leaf, but they restrict it to the upper and the lower epidermis. So they can take it up to the leaf and then basically translocate it all the way onto the epidermal cells and then dump it into the vacuoles that are found in the epidermal cells. This is a stain that basically stains where the nickel is, and that's the epidermal layer. Nickel is also ending up in Iran. There's some research shows that nickel in some of the Iranian plants end up in the trichomes, in the glandular tips of some of these kind of trichomes, which is also found on the surfaces of leaves.
It's kind of a mineral sunscreen.
It's a mineral sunscreen. Well, there's multiple hypotheses as to why plants might be doing this, but they seem to be enriching it on the skin of the plant, right? There are only 2 in California, and I think that's because we just haven't looked. Right? We have 2 in the Brassicaceae. One's a serpentine endemic, Streptanthus polygaloides, which also happens to be a California endemic. Claspi montanum, now Noscia fenderi, which is northern California, southern Oregon, also a serpentine endemic, even though occasionally you find it off serpentine, also take up nickel. Both these plants put their nickel into their leaves. Okay? And in most cases, the epidermal layer as well. So for people who like to hike, and you like to hike on serpentine, I would recommend you buy a pile of filter paper, and then you go to some lab, maybe at San Jose State or Stanford or someplace, get DMG, dimethylglyoxime. You just need a 0.1% solution in water. You basically then impregnate the filter paper in this solution, you let it dry, you put it in a Ziploc bag, put it in your backpack, and then go on your hikes. And whenever you get on a serpentine outcrop, you take this filter paper out, especially if you come across Asteraceae, Caryophyllaceae, Brassicaceae plants, which we have a lot of, crush a leaf, and if the leaf, the filter paper turns red or pink, we know that it at least has 0.1% nickel. It's a really good field indicator of nickel in the field. You don't have to pay $35 to send it to a lab, you don't have to collect leaf tissue, crush it. And if you're not colorblind, unlike me, you will see that color change. Right, so I always have to take students with me because I don't see red well, and so I always get confused between green and red, and that's not good for a botanist. But I've survived so far. Right? So if this turns red, you know that you have on serpentine. So that's how you find nickel on serpentine. How do you know that you are on a serpentine outcrop? I mean, other than obviously kind of knowing it's like a rock outcrop, greenish rock, shiny, not much growing on it, you know that you're on serpentine. But you know the best way to figure out serpentine? This is what a NASA scientist showed me recently. You carry, it's $1.99 on Amazon, right? You carry a magnet pen with you, right? And then, if you're on a serpentinite or ultramafic rock outcrop body, you can pick up serpentine rocks.
Wow.
High levels of iron, magnetite in them, so I use this all the time now because not all serpentinite rocks look the same. In some parts of the world they're red and they don't look anything like the serpentine of California, but it will have the magnetic pull. That's why compasses don't necessarily work on serpentine outcrops, right? So magnetite, you can easily pick this up. So now you know how you get on a serpentine outcrop and you can be sure it's serpentine. Then you open your backpack, you have a Ziploc bag with this stuff. You will find a nickel hyperaccumulator, believe me, we just haven't looked. We need to get out there and do more of it. So get a pen, $1.99. Couple of questions here.
Okay, so the nickel is concentrated in some of the tissues in the upper leaves and so forth. What that means is being denuded from somewhere under the plant. The nickel's being taken up and stored up there, so
exactly.
now it's depleted or denuded down.
It's a cation, so it's still going to move in when there's water. The plant, it's just like potassium or sodium.
Yeah.
As the plants take it up, because plant is growing on serpentinite rock that has nickel in it.
Yes.
So over time, the nickel is going to get weathered into the soil,
Yeah.
and it's going to become plant available, especially if the pH conditions are right, you will get nickel coming into the soil. But we'll get to that. I mean, it's a good question because this has led to a technology that I'm going to talk about later with respect to using plants to clean up nickel.
Yeah.
So why are we only interested in nickel? We're interested in the other heavy metals too, right?
Yeah, it's just that we have nickel is the most abundant heavy metal. It's got economic value associated with it. Chromium is another one, but there are only about 50 species worldwide that take up. Chromium is much more toxic, and there's nothing. So nickel gets into the plant using zinc, magnesium uptake system. So there are other ions that look like nickel, so nickel can compete for those uptake sites. So nickel gets into plants more than other elements do, right? But you're absolutely right. There's other things that we should be worried about too. There's people working on copper and zinc. I'm not talking about all of the metals. I'm pointing to nickel because nickel seems to be the most abundant metal that plants take up in serpentine soils. So leaves is one place. I'm going to get back to your question because I'm going to talk a little bit about it later on. Another place that people are finding nickel, especially in trees in New Caledonia and in Malaysia, nickel ends up in the phloem tissue. This is the phloem, right? So phloem is where the sugar is transported through. This is a bunch of cells that transport sugar. This is a little capillary tube going into the phloem, and the phloem sap is enriched with nickel. Up to 17% nickel in the phloem sap. So there's a theme here, right? You're putting it on your skin, you're putting it in your latex, you're putting it in the most valuable tissue you have in your body, which is the sugar transporting tissue, right? So if you're an insect, if you are something that is going to be eating you, you're going after that sugar. Now you're enriching that sugar with a heavy metal that could be potentially toxic, right? So this is a recent finding from the last few years showing that in trees, nickel does not end up in leaves that are up on the canopy, they end up up and down the phloem tissue. So the idea that has been most heavily tested experimentally is the idea of metal defense hypothesis. The idea is that plants that grow on serpentine soils are challenged of nutrient challenges. There's not a lot of plants that grow on serpentine outcrops. You are basically open prey for anything that goes across this outcrop. Right? You're the only thing that's there, so you may have heavy predatory pressure or pathogen pressure when you are growing on a serpentine soil. So that might be why plants enrich themselves with toxic heavy metals to deter pathogens and pests. There are experimental testing of this hypothesis across the world, and I would say about 80% of the time in laboratory settings, they are able to show that metals deter pathogens and pests and small herbivores from eating the plant. But every trait has there's always pressure for coevolution, right? You come up with some kind of a strategy, something's going to counter it. So here's a plant in South Africa, here's a plant in California. Both of them take up pretty high levels of nickel, 1 to 5%, 3 to 10%. One is in the sunflower family, the other one is in the mustard family. Both of these guys have insects that are only found on these two plants. They are monophagous, meaning they specialize on this plant. The larvae feed on it, they feed on it, they live on it, they lay their eggs on this plant as well. So they are monophagous, complete life cycle over here. This guy spends all of his life cycle on that plant, which is a South African endemic on serpentine soils. They do have different physiologies though. This guy is able to send out everything it eats, even though it has up to 10% nickel. If you look at the body tissue, there's no nickel in the body. Everything comes out. It's able to pass it through its gut, and when you look at the sketch or the waste, that has all of the nickel that the insect just ate. This guy in the Sierras actually bioaccumulates nickel, right? So they have different ways of dealing with it, but they are both specialist insects. They're called nickel insects that grow on nickel hyperaccumulating plants that happen to be endemic to serpentine soils, one in California, one in South Africa. There are other reasons as to why plants may be taking up heavy metals. One is this idea of elemental allelopathy. It's different from the allelopathy idea that you may have heard of, because allelopathy that we generally hear of has to do with organic compounds. Here, what the hypothesis has to do with is that plants may be taking up nickel from deeper levels of soil and basically putting it in the leaves or fruits or twigs, they then drop down to the base of the tree, and that creates kind of a really toxic area enriched with heavy metals right underneath the plant that might then maybe deter or restrict kind of germination of other plants right underneath it. It's been tested to like 50% of the time they're able to show that this is going on, 50% of the time they haven't been able to show elemental allelopathy. Here's another idea that's out there, is the idea that nickel is a cation. It's a divalent cation. It's just like sodium or potassium. It could be used to adjust the osmotic conditions in the leaf. So that if you're growing in a soil that lacks some of these cations that plants generally use to adjust their osmotic conditions. The reason that a plant that grows on seawater, in seawater or near the coast, is that it can make itself saltier than seawater. That's how water comes in, right? So in this case, maybe these plants are enriching themselves with nickel that is abundant to make themselves saltier so that water can come into the plant in an environment where water is hard to get. This is another hypothesis that is being tested. We're actually testing this hypothesis right now at Cal Poly using Streptanthus polygaloides from the Sierras. This is, I think, the hypothesis that I think is really exciting because it also contributes to evolutionary thinking. The idea that pollinators can detect heavy metals in nectar, so plants might be enriching their nectar with heavy metals. That could then eventually lead to some kind of a pollinator tolerance preference idea that might lead to isolation, right? If pollinators can only go to, if pollinators are tolerant of heavy metals, they'll visit those plants that are on the serpentine outcrop, stay away from the serpentine, plants that are off the serpentine soil, and eventually that might lead to some kind of an isolation that could lead to an evolution of new species. So finally, I wanted to kind of talk about this really cool technology. I just came back from Albania, where farmers are now moving away, going from growing traditional crops, especially in areas where they live next to old mines, nickel mines and chromium mines. And they are now growing plants, both in South Africa and in Albania and Greece and southern Spain and parts of France. They're growing basically metal hyperaccumulating plants as crops, right? So, and they're harvesting biomaterial that has metal in it that can then be used, processed to extract metal out that can be then resold into whatever we need metals for, right? So this is some photos that I took in Albania where large, 100, 200 hectares of land is being converted to metal farms. And traditional farmers are now moving into this area. The material is bought by a company in southern France and parts of Spain, and they basically move this material there, process it. And the idea here is that you treat these metal hyperaccumulating plants as a crop, you water it, you treat the soil, the plants take up the heavy metals, put it into leaf tissue, you harvest it, then you basically ash it, treat it chemically, and then you end up with bio-OO that essentially can be sold. And you can use this in places where the level of metal is not high enough for traditional methods to be useful, right? And it's of course much more environmentally friendly than trying to take tractors and bulldozers and strip mine outcrops, right? So again, the idea is ultramafic soil, serpentine soil, a brassicaceae plant or an asteraceae plant. Then you basically ash it, you end up with bio-O, then you treat it to get the pure salt out that can then be converted into the metal that can be sold for whatever you need nickel for, right? This technology now exists for gold, it now exists for nickel, for copper, zinc, and to some degree chromium. It's still not, except for nickel and gold, it's still not economically viable. And the techniques have some caveats to pay attention to. I'll talk about them in a bit. But it is catching on. Not in the US, but in Europe and parts of Australia and Africa. People are looking into this as a kind of a viable way to clean up contaminated soil and at the same time make some extra money out of it. Right. So here's my friend Alan Baker, who's now retired from University of Melbourne. And he's holding about 70,000 Australian dollars worth of gold in his hand. takes care of 2 technician salaries or 3 graduate student salaries from gold extracted using Brassica jansea, which is a plant that is naturalized. It's originally native to India and parts of South Asia, naturalized across the world. It's found here in California as well. And it is a plant that can take up huge amounts of gold into the leaf tissue. But the problem with this technology is that the gold does not get into plant unless you add ammonium thiocyanate to the soil. So you're adding cyanide to the soil to make the gold come into the plant, right? So they do this in kind of swimming pools. Old swimming pools are tanks where the soil can then be disposed of in an environmentally sensitive way. But now there's some technology coming out from France where they have found another chemical that can be used instead of cyanide to make gold bioavailable. So there is some issue with this. This using for nickel is that you don't have to do anything really. You might have to adjust the pH of the soil by adding something that would acidify the soil a little more so that heavy metals become more bioavailable so the plants can take it up. But this is a much more environmentally friendly, easy thing to do than working with gold. If you're interested in this technology, I can send you some really cool papers. A lot of it's coming from Southern Europe, Mediterranean Europe, and there's a lot of interesting technology now associated with phytomining.
Rare earth metals are kind of becoming globally a strategic resource for military and high tech and stuff. And they're not everywhere that, not all nations possess these resources.
I'll send you a review on rare metals. There are some plants, so people are looking at the cell phone stuff, things that get into cell phones, whether there are plants that can naturally take up these heavy metals. And there are, for some of these rare metals you're talking about, there are some plants, mostly in the Brassicaceae. The Brassicaceae is amazing. Not only does it give us cabbage and broccoli and cauliflower and kale and kohlrabi and brocoflower and all of that stuff, which also happens to be the same species, but it also has these really interesting plants that have this really unique physiology that enables them to take up heavy metals in this case. But yeah, give me your email. There's a couple of papers I've seen recently that talks about some of these rare earth elements that are hard to get, expensive, and where the plants might be a useful kind of way to extract some of these heavy metals from the soil. So there are other, there are other reasons why this technology is taking off. Organic nutraceuticals, you don't have to take, you can use some of these things, organic material that are enriched with things that we need in our body, right? These are things that we need. We take multi-mineral pills, so why not take a little leaf of Alyssa murale instead, right? That has all of that stuff. It's green, so it's good for you, and it's got some heavy metals in it, as long as you know what the concentrations are. Trace element supplementation, zinc, selenium. Selenium is lacking in cattle, for example, so you could feed cattle with plants that have heavy metals in them. Trace element crop fertilization. Nickel is a micronutrient plants do require it for growth. Zinc is another one. There's a lot of Class P or Alyssum species that take up zinc into their leaf tissue, so they could be used as crop fertilization as well to limit the amount of the usage of chemical fertilizers out there. There are concerns with this technology. We shouldn't be running with it just because it sounds really cool, because we are facilitating the spread of heavy metals into the environment, right? So we are making these metals more bioavailable. Plants take it up. We are growing them in large, fields. So, they're facilitating the spread of these heavy metals to the next trophic level, by making this much more bioavailable. There's a really cool video. I can just send it to whoever sends me an email. I have my email later on. This is a plant that was brought from Mediterranean Europe. Have you heard about this Alyssa murale problem in southern Oregon, northern California? It's a Brassicaceae plant that was brought from Europe. It's a serpentine endemic, and we bring it to part of the US where there are lots of serpentine. And I remember I was a graduate student at this conference where the USDA was proposing this as a something that they were going to do with all the old nickel mines up and down Oregon and California. And I asked the question as to whether they have any concerns about this thing escaping. They're like, oh, no, it's not going to happen because it's only on serpentine. But I said, well, there's a ton of serpentine there. So basically what happened was there was a company called Viridian, which was using this to extract heavy metals. They used Alyssum to grow this, and then the company just somehow after the operations ended, they just left all the plants on one side. So it's become an invasive species up in southern Oregon and northern California. It's moving on to serpentine outcrops. And people are surprised. I mean, why would you be surprised? It's serpentine endemic in another Mediterranean climate. You bring it over here, obviously it's going to take off, right? So there's a really half-an-hour video on it. If you're interested, I can send that to you, talking about invasive, as things you have to be concerned about when you're thinking about this technology. Soil amendments, you don't want to be adding cyanide to the soil because that's going to hurt us more than help us make money. There's also a huge push to genetically modify plants to make them take up multi-elements, not just nickel, but why not take up 20 metals at the same time, and why not make this plant a poplar? That will be fast growing, right? So this is currently happening. There's a lot of genetic modification work going on of transferring these brassicaceae genes into larger, fast growing trees that can then be extracting large amounts of heavy metals from the soil, and that we should really walk very carefully in that kind of area, because we are not only making things, we are essentially making the facilitation of heavy metals into other trophic levels much easy by doing that, right? So, all of these cool things about serpentine plants and rock outcrop plants, to be able to continue to learn from them, whether it's ecology, whether it's finding of endemic species, rare plants, all these cross-kingdom interactions I'm talking about. I didn't even talk about mycorrhizae, I didn't talk about bryophytes, I didn't talk about lichens. There's a lot of cool stuff. We just described 2 new genera of lichens from Serpentine in South Africa. Like completely new genera, never been discovered before. And that is not because we were really good, it's just because no one's really studied lichens of South Africa since 1957. Right? So we go there and we find all these new species on serpentines of South Africa. So to be able to continue this kind of study of rock outcrop plants, we've got to also face the challenges these plants are experiencing. So one of the big challenges, like everything else for biodiversity, is climate change, right? So I mentioned this earlier, the idea that you really have to know about substrate affinities of these organisms to understand their potential to succeed or be able to kind of make it in a rapidly changing world. Right? You've got to understand that so we can have better kind of conservation plans associated with either restoration or conservation of some of these species. So here's an example of coming back to the earlier slide that we showed. This was taken by my advisor at University of British Columbia in the early 80s. And here is this beautiful display of Laciniia up on the Serpentine, the grasses on top. I don't know what it looks like because I haven't been there since 2009. 2009 it looked like this. Right? So 1980, 2009, is it any better? Is things not looking good, right? So part of the reason that this might be happening is because there is a issue of aerial deposition of nitrogen that's happening across the Bay Area. We know from Stu Weiss's work that this is really happening up on Kirby Canyon along Highway 101. There's high levels of nitrogen, 11 to 16 kilograms of nitrogen dumped by rain on these nutrient-poor habitats where plants have adapted to live with nutrient-poor conditions. When nutrient levels go up, what happens? All of your friends, or not so friends, end up encroaching these serpentine soils because they can thrive under these high nutrient conditions, right? They can outcompete these slow-growing serpentine endemics or plants that are native plants that are kind of adapted to deal with these nutrient-poor conditions. So this is happening globally. At Harvard Forest, they're showing that pitcher plants of the genus Sarracenia are not making as many pitchers that they used to 30 years ago. So the plants still survive. They're still there. The populations are as dense, but there's a higher degree of non-pitcher making plants in these bogs. And part of that is also bogs are also becoming nitrogen enriched. Right? So why make a pitcher? So there could be selection driving those traits from those populations as nitrogen levels go up. When we think about climate change, we are thinking about the carbon footprint, we are thinking about global warming. We're not really thinking as much about the chemical changes that change the soil environment that could have a huge impact on populations of plants that are restricted to unusual soils. Of course, this is Stu Weiss's work and Paul Ehrlich's work showing that with this kind of invasion of grasses, you could have plants like Plantagos and Castellias that the bay checker spot butterfly relies on for its larvae to feed. They get kind of outcompeted. That might be contributing to kind of the population kind of crisis that some of these insects are facing as well. And this is happening on limestone in Europe. It's happening in parts of South Africa. Some of these insects are disappearing from harsh rock outcrop communities because their plants are disappearing. In the East Coast, in Maryland and parts of Pennsylvania, a big problem has to do with suppression of fire and grazing, right? That has led to the invasion of serpentine barrens of Maryland and parts of Pennsylvania and North Carolina and Virginia of invasive pines that are coming in and basically acidifying the soil and basically taking over some of these outcrops that has quite a lot of, not endemics, but largely restricted species that are native to these grasslands of the East Coast. So suppression of fire, suppression of grazing also contributes to the invasion of these serpentine outcrops. So if you want to know more about serpentine and rock outcrop plant communities, I encourage you to join the International Serpentine Ecology Society that was founded in California in 1991. Every 3 to 4 years, people get together in a serpentine hotspot, New Caledonia, South Africa, Cuba, Italy, Albania, Malaysia, Portugal. The only reason that we got together in Maine is because I was teaching there, so I brought them over to Maine. But I did end up taking them to Gaspé Peninsula in Mount Albert in Quebec. There's a lot of interesting serpentine, alpine serpentine that you get up there. But we also had one in Maine. So all of these conferences lead to proceedings that are peer-reviewed, published in journals or books. The first one was in a book, the one out of California. Everything else came in a regional journal in that part of the country where the conference was held. And then the next conference happens next June, 2020. 2020 is going to be a big year, right? One, because of the elections. Second, because there's a conference in Russia, of all places, right? So it's going to be in Russia in 2020, in the southern Urals, right? If you have it's not, the ticket from San Francisco to Yekaterinburg is anywhere between $900 to $1,200 right now. Right? So, if you want a break and want to go to Russia in 2020, this is where you want to go. You want to go there, and if you want more information, send me an email, my first initial last name @gmail.com. I'll put you on the mailing list so you can meet 300 of us who are crazy about serpentine. So my plan is to propose Cal Poly as the 11th venue for the international conference. And Cal Poly, obviously, area is really great. We have really good student housing where delegates can stay. A lot of good conference facilities, tons and tons of serpentine. There's serpentine on campus. So when people ask me why I left San Jose State, I'm like, there's serpentine on the Cal Poly campus. I can just walk to a serpentine outcrop. I don't have to drive to a serpentine outcrop. So for those reasons, so California Botanical Society had their meeting at Cal Poly, and I took them over to a serpentine outcrop. So this is the group that joined us from across California, mostly grad students. But that's going to be in 2023. So put it down on your calendars if you want to think that far ahead. And Cal Poly area is really great. We have a lot of serpentine endemics. There's a bunch of them have a specific epithet, Obispoensis or Obispoens, right? It's from San Luis Obispo. A lot of these things are largely restricted to serpentine. We have Sergeant Cypress, which is one of the southernmost populations up on Cuesta West. You have a large stand of Sergeant Cypress, which is on serpentine, just about 10 minutes from the Cal Poly campus. So think about it, 2023, but before that, 2020, Russia, if you're interested on serpentine, and thinking about some of these things that I've talked about. So with that, I want to kind of remind you that when you see these kinds of rock outcrops or barrens, when you go to New Italy, just don't think of it as a wasteland. These are unusual habitats that have a lot of specialized plants that happen to be rare, endemic. They're model systems to ask key questions in ecology, evolution, conservation. You can do research all the way from the cellular level to ecosystem level using these plants and these rock outcrops as model habitats. So with that, thank you for your attention, and thank you again for the invitation.